This lecture, part 4 of 4, will cover the structure and functions of the cell. The topics of protein synthesis, the cell life cycle, and cell differentiation will be discussed. DNA is found in the nucleus of a cell and contains the genetic information. The DNA controls the function of the cell by providing directions for protein synthesis from its genes. The process of protein synthesis involves both transcription and translation. Transcription refers to the process of RNA production from DNA and takes place in the nucleus of the cell. Translation refers to the process of protein construction by the ribosomes, which use the information contained in the RNA produced from DNA in the process of transcription as a blueprint. This takes place in the cytoplasm of the cell. Let's examine the details of transcription. Again, this process will occur in the cell's nucleus. Recall that DNA is composed of two strands, twisted into a double helix structure. In transcription, an enzyme known as RNA polymerase binds to a promoter of a gene found in the DNA. A promoter segment marks the start of the gene. Each triplet thereafter codes for a specific amino acid that will be part of the protein. So here we see the RNA polymerase, this structure in blue here. It'll go ahead and bind to that promoter region, the start of the gene that's going to hold the genetic information that codes for the different amino acids that should compose the protein. Each set of 3 bases, here, 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 etc., is known as a triplet, and each triplet codes for that specific amino acid that should be inserted to the protein sequence at that point. As the RNA polymerase travels the strand of DNA, a strand of messenger RNA is produced. The messenger RNA will be composed of a sequence of bases that is complementary to the base sequence found on the strand of DNA. That is demonstrated here under number 2. So here we see the strand of messenger RNA. This is the strand of messenger RNA that's forming. What will happen is RNA nucleotides that complement the nucleotides found on the strand of DNA will be brought and added to the strand of messenger RNA. So in this case, we have a cytosine on the DNA, and we will bring that RNA nucleotide, in this case, a guanine, that'll go ahead and correspond to that. This will become the next nucleotide in our sequence on the messenger RNA strand. It is important to note that RNA will use uracil instead of thymine as a complement to adenine. So we'll see that going on here. Here we see an adenine on that strand of DNA. So instead of a thymine, we're working with RNA, remember RNA here, we're going to go ahead and bring that uracil. That uracil will be its complement. So we'll have adenine complementing with the uracil on the messenger RNA. The messenger RNA will continue to form until the RNA polymerase reaches a stop codon, seen here, which indicates the end of the synthesis of the strand of messenger RNA. At this point, the DNA strands will reattach, and a single strand of that messenger RNA has been formed, holding a translated copy of the genetic information for a specific protein. Transcription is necessary because DNA cannot leave the nucleus of the cell to reach the ribosomes, where protein synthesis takes place. However, messenger RNA formed through transcription can. This is how the DNA can communicate its genetic information or instructions with areas of the cell outside of the nucleus. Once the messenger RNA has been synthesized, it will leave the nucleus via the nuclear pores of the nuclear envelope and head off to the ribosomes in the cytoplasm, where protein synthesis will take place. The process of protein synthesis here is going to be known as translation. Translation will begin with the binding of the start codon to a small ribosomal subunit. So we have our messenger RNA leaving through one of those nuclear pores headed to the cytoplasm. Once it reaches the cytoplasm, that messenger RNA is going to go ahead and attach to a small ribosomal subunit with what is called its start codon. Okay? Start codons always code for the amino acid methionine. Translation will begin after this start codon has bound to the small ribosomal unit. Each codon, or set of 3 bases, will have a corresponding anticodon located on what is called a transfer RNA. So the transfer RNA is going to be this molecule here, this compound, okay? Has a characteristic shape, kind of like a hairpin shape there. On the transfer RNA, we're going to have what is called an anticodon, which if we take a close look at, we see is actually complementary to the start codon. We have an A with a U with an A, and a G with a C. So those bases there are going to be complementary pairs, which is going to attract that specific transfer RNA to this region. The other important thing to note about RNA is that it's going to carry an amino acid with it. The amino acid that it carries is going to be specific to the anticodon found on the transfer RNA. So the codon on the messenger RNA is going to attract a specific anticodon on transfer RNA that holds a specific amino acid. And that is how the genetic information is able to be translated into an amino acid sequence, hence the process of translation. After this begins, a large ribosomal subunit will join up with a small ribosomal subunit and enclose the messenger RNA, as we see here. The next transfer RNA arrives and the anticodon binds to the codon of the messenger RNA, bringing 2 amino acids in close proximity to each other. A peptide bond will form between the 2 amino acid, and the first transfer RNA will leaving its amino acid bonded to the amino acid on the second transfer RNA behind. So we'll go ahead, form that peptide bond. The first transfer RNA that was there will go ahead and leave. This ribosomal unit will continue to move down that strand of messenger RNA. And the result here is we have a peptide bond between 2 amino acids. Now we've freed up room here for the next codon to go ahead and attract the anticodon found on another transfer RNA molecule, and a third amino acid will be brought into the mix. This ribosomal unit will continue to move down the messenger RNA, and the process of adding more amino acids through those peptide bonds will continue, developing a polypeptide chain. Once a stop codon is reached, the synthesis is complete and the components will part ways. The result is a polypeptide chain, or a protein if the chain is longer. This protein will be an expression of the genetic material found in the DNA of the nucleus, and the newly formed protein will go on to do specific functions in the cell. So this is how the DNA is able to communicate its genetic material to the cytoplasm of the cell, specifically in the ribosomes, where we're going to be able to make these specific protons based on the instructions from the genetic material. Each of these proteins that's produced will have unique functions, and these unique functions will then be demonstrated in the cell, giving the cell unique characteristics and functions, all as a direct result of the control of our genetic material, the DNA. This table gives some examples of that triplet code of those bases. So here we go ahead and see we have a triplet for DNA. The messenger codon, or the messenger RNA codon, is going to complement that. So in this case, we had 3 adenines. Now we'll have 3 uracils. The set of 3 uracils is going to attract the anticodon of 3 adenines, the AAA, on that transfer RNA. And as a result, this specific transfer RNA with this anticodon is going to carry along with it a specific amino acid. in this case phenylalanine. So this is how that triplet code codes for those specific amino acids and how we're able to go ahead and build that specific sequence of amino acids in the protein. Now let's switch gears and take a look at the stages of a cell's life cycle. The purpose of cell division is the growing of new somatic cells and the replacing of old. For this to occur successfully, the genetic material needs to be copied correctly and correctly distributed to the forming daughter cells. Mitosis is the DNA replication and nuclear division of the genetic material in the nucleus. Meiosis is the production of the sex cells, the sperm and the ova. And apoptosis is the genetically controlled cell death for cells that do not divide, examples being infected or abnormal cells. This diagram represents the stages of a cell's life cycle. Most of the life cycle is spent in interphase, the time between cell divisions. This is the time of a cell's life where it carries out its normal everyday functions. As a cell gets ready to divide, it will enter the G1, S, and G2 phases of interphase. In the G1 phase, normal cell function continues, and extra organelles and cytosol are made in the anticipation of the upcoming cell division. In the S phase, DNA replication occurs as well as histone synthesis. This slide demonstrates how DNA is replicated. The strands of DNA separate, and an enzyme known as DNA polymerase forms a complementary strand of DNA nucleotides. The result is two identical copies of the original DNA. After the S phase and that DNA replication, the G2 phase of interphase is reached. Here, centrioles are replicated and some protein synthesis occurs. The cell is then ready to enter the stage of mitosis. Mitosis consists of the stages prophase, metaphase, anaphase, and telophase. So, at the end of interphase, the cell has prepared itself for dividing by making a copy of DNA and also extra cytosol, proteins, and organelles. The next phase will be prophase. In prophase, DNA tightly coils and can be seen under a microscope. A centromere connects the two sister chromatids. The nuclear envelope disappears and the chromatids attach to what are known as spindle fibers. So here we see the centrioles. Okay. We're going to see that the chromosomes appear and we have centromeres that hold the two sister chromatids together, and they're going to attach to what are called spindle fibers, these long strands that are filling the center region of this cell here. So we're going to go ahead and have those centromeres with those sister chromatids attach again to those spindle fibers. In metaphase, the chromosomes move and line up along the center of the cell in a region known as the metaphase plate, seen here. This is followed by anaphase. During anaphase, the chromatid pair separates and the daughter chromosomes are pulled to opposite ends of the cell. So we have each of these daughter chromosomes being pulled in opposite directions to the opposite ends of the cells. This leads to telophase, where the nuclear membrane forms, the nuclei enlarge, and the DNA begins to uncoil. A cleavage furrow can be seen. This ends mitosis. However, the process of cytokinesis, the cytoplasmic division that forms the two daughter cells, officially marks the end of cell division. Cytokinesis begins in late anaphase and is complete when the nuclear membrane of the daughter cells have formed. It is important to understand that all somatic cells in the body have the same chromosomes and genes. They have the same DNA. Yet, they develop to form many different types of cells. This occurs because of something known as cell differentiation. Cell differentiation takes place when specific genes are turned off, leaving the cell with limited capabilities. Because not all of the genes are expressed, each cell type will have unique characteristics. And a collection of these cells with unique characteristics and functions is known as a tissue. Here again, we see the diversity of cells found in the human body. Let's review with a checkpoint. How does the nucleus control the cell's activities? Feel free to pause the lecture while you formulate your response. The nucleus uses DNA to control the cell activities. The DNA codes for proteins, and depending on what proteins are created as genes are expressed, the cell will have different characteristics. Also, DNA can order specific things such as enzymes to be made, which can then alter the activity of a cell based on the conditions that are present. Let's go ahead and try one more. Can you define mitosis? What are the 4 stages of mitosis? Mitosis is a process found in cell division that results in a single nucleus of a cell dividing to produce 2 identical daughter cell nuclei. The 4 stages of mitosis are prophase, metaphase, anaphase, and telophase.